Okay, Assalamualaikum, Bismillahirrahmanirrahim, Assalamualaikum, and a very good afternoon. I bid to all participants and welcome to our INBIOSIS webinar lecture series number 9 for year 2021. I am Dr. Nurkhalida Kamal and I will be the moderator for today's. I hope the video that we showed earlier has provided you with some background about INBIOSIS and also various services that we offer. If you're interested to collaborate with us, or if you would like to know more about our services, please visit our I'm very sorry for some disturbance, so I will continue. So basically, INBIOSIS webinar series is an established platform for researchers and their significant knowledge and research experience. Despite with the current pandemic situation, I hope all of us still motivated and connected. Now, everyone, with some housekeeping rules for our webinar today. The lecture will take about 30 minutes and the Q&A session will take at least about 15, maybe to 30 minutes. If you wish to ask the presenter a question, please submit the questions either in the Zoom chat box or in the comment on our Facebook page. Inshallah, we'll read all questions during the Q&A sessions. And before the seminar ends, I would like to invite all of participants on the camera and for a photography session. And please stay tuned until the end of the session to fill in the attendance form for the ticket and also for ISPAL UKM credit. A brief introduction about our speaker of the day, Dr. Ahmed Tawfike. Doctor is a pharmacy graduate with keen interest in drug discovery. He successfully achieved a master's degree in analytical and natural chemistry and a PhD in metabolomics from the Strathclyde Institute of Pharmacy and Biomedical Sciences in Glasgow, UK. Had a couple of postdoctoral positions at the Strathclyde Institute and Bangor Universities before he joined Rodden State Research as a metabologist in the Molecular Discovery Group. Ahmed is currently a senior scientist at Owlstone Medical in Cambridge, developing breath biomarkers which could be relevant to a number of life-threatening diseases like lung cancer and hepatic cirrhosis. Ahmed has an excellent expertise in analytical chemistry, metabolomics, chemometrics, and machine learning approach in metabolic profiling. He has strong analytical skills in the data-driven therapeutic discovery, features extraction, biomarker discoveries, and compound characterization. Ahmed was appointed third evaluator at the European Commission for EU Horizon 2020 proposals. He is also an editor for Simental and a reviewer for various journals, for example, diagnostics, metabolites, molecules, marine drugs, fungi, microorganisms, as well as ACS Omega, Journal of Chemistry and Biodiversity, and scientific report. I would now like to welcome our speaker for today, Ahmed. Please welcome Dr. Ahmed.
Right, okay. I would like to, first of all, I would like to thank you, Dr. Khalida, for the introduction, and I thank the Department of System Biology and the National University of Malaysia for inviting me for this webinar. I'm very delighted to present my previous experiences and knowledge yeah, for you all, for probably further questions. And I would like to start immediately, just for sake of time. So I will share my screen. Right. So does everybody see my screen now? Yeah. Okay.
Yes, Ahmed.
Right. Okay. So yeah, just so I'm actually going to speak today about the of metabolomics in natural product chemistry or natural product drug discovery. And to start, so I would like actually to show first what kind of application actually are my experiences. And before I show that, which mainly two application, drug discovery and biomarker discovery. So I would like just to notify you that I'm using untargeted metabolomic analysis, which I am looking for everything. I mean, like all metabolites, known and unknown. And I identify a sample subject, select optimal separation and universal detection strategy to maximize the data, extraction, sensitivity, reproducibility. And then I use chemometric method and chemical database to identify significant biomarker. In case of biomarker discovery, in case of drug discovery, I need to use like some other additional cohort like chromatographic exploration and stuff like that to isolate and purify the compound, and then use NMR, two-dimension NMR, and MSMS for the identification of these compounds. So mainly in the drug discovery section, I worked on plants and the plants themselves, the plant, and I worked also on marine bacteria. And the main aim here was actually is finding some molecules which could be relevant to a biological activity or mediating for the biological activity of a plant or bacterial or fungal extracts, and actually how these metabolites could be changed according to the gross condition. For example, if we use the culture condition, if we actually doing fungal or bacterial culture and changing media, changing the gross conditions, so how that would affect our bioactive molecules. So the things actually which I have worked on: anti-cancer, antioxidant, antimicrobial, specifically anti-trypanosome molecules. And as I told you, here in the drug discovery application, if something is actually proven to be a new or novel compound, so we need actually, and this is actually the metabolomics actually would tell us if this compound is novel or molecule. So I need just to chromatographically separate and elucidate these compounds. In the biomarker discovery section, so I'm looking for a specialized molecules, and these molecules could be bioactive molecules, could be actually molecules relevant to a biological status or physiological status, phenotypic trait, environmental. So yeah, any molecules could contribute to any of those, and it could be relevant to something we are interested in. We use the metabolomics actually to identify or to highlight this biomarker. And yeah, I use several chemometric methods in this, like all the multivariate data analysis and molecular networking for actually discovering such biomarkers. So I will move now to the workflow. So the workflow actually start, so this is mainly the untargeted metabolomic workflow. So start with sample preparation. So I worked on different samples, plant, bacterial, fungal, and some biological samples as well. And actually the instruments I've used, NMR, LCMS, GCMS, for sample analysis and data acquisition. And then the output of this instrument or the data coming from this instrument is immediately going to the data processing step. So in the data processing step, there's two main steps, which is like feature extractions and function annotation. The feature extraction one, which is actually involving several processes or steps like baseline correction, removing noise, big detection, deconvolution, big alignment, normalization. So you're trying actually to come up with a feature table with aligned feature table. Everything is aligned in one table, so you can compare the sample or the molecule all over your samples. And the second thing in the data processing is a functional annotation, which actually is the integration of the spectroscopic data coming from the instrument with metadata. The metadata could be biology, could be soil data, could be taxonomic data, could be location, could be gross condition, could be anything which could be relevant to this spectroscopic data. And we need to find somehow correlation between both two. So two independent data, and we need to find a correlation between them. So, and this is send us to the next step, which is the data analysis. So in which our chemometric method, as I said in the previous slide, so supervised and unsupervised method like principal component analysis and OBLSDAs. Then I also, so this is actually the purpose of the data analysis step, is to find classes, cluster or trends in my data, and also actually just to be able to highlight some biomarkers through the supervised multivariate data analysis. After in between, actually, there's a metabolite identification. So when I see some interesting molecules which could be relevant to any of a specific status or biological activity, I actually try to find out actually if these molecules are something known or unknown. So I search database like Dictionary of Natural Product for the natural product stuff, and human metabolite database for human metabolites. And then for some biological interpretation, sometimes I use correlation networks or pathway analysis. So this is actually the whole thing. And I will actually give you some examples from my previous work for you to see how really I employ these methods into my work. So the first of all, like this slide, actually, as every natural product chemist, I start from scratch, from preparation of the extracts. This is actually our bacterial extracts, liquid media, and then we just extract these flasks and make like an extract, this extract, and then actually I just try to screen to see, or just run this over the LCMS to see actually how that looks like, if there's any interesting peaks or something. And then actually if I find and I do some dereplication for these actually extracts, just to see actually if there is something interesting in there. Some literature review to see actually if there is a biology or biological activity for these molecules, and whether these molecules could be new or something known from before. And then when I decide, oh yeah, I need just to further do chromatographic separation, so I go to the flash chromatography, and as you can see, this is actually the instruments we used in Stride in Scotland. It's C18 column, and you know, actually the trend, which is like reversed phase columns. So you run like a water acetonitrile with 0.01% formic acid, and then actually the fractions could be pooled together using like a UV, ELC chromatogram, and TLCs as well. So as you can see, this is the chromatogram just coming out from the flash chromatography. You can see a group of metabolites that we are able to identify. It's like polar to semi-polar metabolites, and on the other side, chromatogram, which is this one, you could see actually some series of hydroxycarboxylic acid, fatty acids, which are actually the one responsible for the biological activity of this bacterial culture. As you can see, there are series of fatty acids, and I use mass spec databases actually just to identify this compound, and you can see here in a mass spec the difference in retention time between the fatty acids. So these fatty acids, if you can see here the exact mass, you would be able to say like, oh, there's a CH2 difference between every molecule. So there is a 14 mass unit difference, and this 14 mass unit make a 2-minute difference on a TIC. So here's this one, 26, 28 minutes, 32 minutes. And actually I used also NMR. Oh yeah, so the NMR is not here now, but I used also NMR for identification of these molecules. So this is basically is a pure natural product chemistry I used to do. But in the other part, which is actually the metabolomic part, then I use metabolomic tools actually just to reach me or to lead me to the conclusion that I need to further chromatography to separate this chromatographically by the bioactive molecules. So for instance here, this is a study is actually made on the British Bluebell. So this is actually made in North Wales in Bangor University. So when I joined them, there was a PhD actually working on the British Bluebell, which is actually was growing in the farm of her supervisor. So she collected actually the British Bluebell all over the growing season. So she had like many samples, and actually she was looking at the carbohydrates. So targeted analysis, targeted metabolomic analysis, actually measuring carbohydrates and see actually how it's different between the concentration of this carbohydrate different between season to another. So when I actually came in, so I suggested that we can do some untargeted analysis on the British Bluebell. And we searched the literature, and we found that these British Bluebell could have some antimicrobial activity, and they are reaching saponin molecules. So the first thing actually I've done is actually a BCA comparison between the active in red and inactive extracts, and trying actually just to find what molecules could be responsible for the activity of the active extracts. So here, so there are, as I said, actually, so the molecules which are actually responsible for the activity of the Bluebell are saponin-like molecules. as you can see here, and some of them was already identified in the database, but also some other are not really or were not really identified in the database. So we decided to move on and go for with a chromatographic separation and purify the bioactive molecules. When we purified the bioactive molecules, so we found a molecule at the center here, which is 1445 Dalton, and this molecule was actually not identified before in the database. So we don't know actually anything about the molecule. So we run a molecular network, as you can see. So the molecular network basically is actually connecting the metabolites which share the same fragmentation pattern. So this is an SMS molecular network. So all molecules that actually correlated together, they share the same fragmentation pattern. So we were able to know now from here that the 1445 is structurally related to the 1283, which was already in the literature identified as this compound. So if you calculate the difference between the two, it was like 160 something, which is actually equivalent to a 1 sugar molecules here. So this new compound is completely new because it's an extra sugar molecules on that end. And we were able to confirm that from the NMR data. So this is how actually basically metabolomics and molecular networking are useful in our work in natural product drug discovery. Another example here, it's basically, this is fungal culture, aspergillus, solid culture, rice solid culture. And this molecular network is actually made out of MS1 mass spec. So it's not MS like the one in the previous slide, but this is only MS1. So these molecules are correlated together because of actually, or by Pearson correlation. If some of you are aware by Pearson correlation, so we try just to find which molecules are upregulated or downregulated together, and these molecules actually be in the same cluster. But one of the things actually I integrated a biological activity into this molecular network, so we could see bio here. So I integrate the bioactivity that we have measured for this extract into this molecular network. I'm trying just to see what molecules could be correlated with this bioactivity. And you can see here, this is giving you the thickness of this an idea about how strong this correlation. So for example, we found one of the strong correlation with this molecule aspergillomarine. So aspergillomarine, this we found that in the database, and we use chemotaxonomic classification to filter out the hits that we found. And this actually lead us to this molecule. So this molecule actually, we tried just to make sure that this, because this is an only MS1 data, so we tried to use another independent identification method to make sure this is the right molecule. So we use NMR here to find, and this is NMR for extract, and this was actually molecules in the extract, and we found that this is a hydroxyl group, for example, and this actually is A, B, C, these aromatic molecules here in NMR. So the structure was identified by searching a dictionary of natural product for the predicted molecular formula, and the hits were reduced by applying chemotaxonomic filter, and the structure was confirmed by proton NMR. So because this is actually a molecule was identified before in the literature, so we didn't need actually just to separate this molecule. We just stop at this point, and we knew that these molecules could be the one which is responsible for the activity that we have seen for this fungi. The same thing actually I've done also for in another one published in Metabolites. It's differences from Euphorbia, and we try actually just to, so these are the different species here, and some of these molecules, so it's really natural that the species, the molecules which actually belong to the same species to actually cluster together, but you can see in the middle here, some of them is actually could be sharing between two species. And also I integrate again the anti-cancer activity against breast cancer cell line and hepatic cancer cell line. And you can see here is some molecules which actually could be relevant or could be correlated with this biological activity or anti-cancer activity. And if you zoom in, there is like a pie chart, and the chart could tell you how much this molecule is present in every of the species. So you can say, for example, this 1338 is present in this one, which is Euphorbia granulata, and this one, the most species actually producing this molecule in a higher capacity, I would say. So this is actually something I've done as well. The same thing is being done also for the tabiboya, and tabiboya is actually one of the plants which belong to family Bignoniaceae, and it's actually very famous with anti-cancer molecules. And I've done the same thing. I was able also to find some interesting structure which is actually correlated with the biological activity. One application I've done in Rothamsted here in England before I just leave them was actually experiment which we are trying just to find some molecules which could be correlated. This is a biomarker discovery application of Metabolomics. So I was trying just to see if there are some molecules could be correlated with the metal toxicity. So sorry. So basically, there was this is a plant, and actually it's been treated with 4 different metals: cadmium, lead, copper, and the green one, which is like this is control, not treated with anything, but actually these are the metals which apply to the soil, and we're trying just to see actually whether this would affect the metabolite or the metabolite fingerprint of this plant or not. So as you can see here, I run a molecular network from the MSNS spectra, and there is also like some facility to use to make like clusters, sorry, or to make classification based on the chemical classes. So I just colored here by chemical classes. So this one actually basically of maybe flavonoids here. And then if you zoom into that one, you could probably see here the pie chart and how these metabolites actually presented in the different sample. You can see this one of the interesting molecules here, the 423, and it's completely and it's actually connected with the 469, which is the adduct, formate adducts of the 423. So they are connected together in the same cluster, and you can see here the difference is 46, which is actually for the formate adduct. And these molecules, if you can see, is represented in the 3 control, and is not really, or very, very, very tiny line here in the control. That means actually this molecule is overexpressed in the metal treated samples more than the control one. That means actually this molecules have has a role in the defense mechanism of the plant against this metal toxicity. So this molecule is completely known to us, but we try, so we try now to see actually whether this, what actually, how much involved these molecules in the defense mechanism, and and what that could be, and how this happened, stuff like that, before we decide to publish the data. So this is actually most of the application I've done, and I wish that something there was something interesting for you guys in these slides. And I wish to thank again everybody who invited me to this presentation. And if you guys have any question, I'm willing to answer. Thank you.
Thank you, Dr. Ahmed, for your very nice and short and very interesting talk. I think everyone can see how does actually metabolomics can be applied in many ways in natural discovery, not only just isolating just a new novel compounds, but it depends on the question that we really want to ask. So just a question, we have several questions, but let's the audience asking the question by typing in the chat box first. So just a very basic question, because maybe some of us really want to start a metabolomics work. And the question is, we need a really, huge spec for the computer specs to start with the metabolomics and molecular modeling, or is it just that we can use it?
So are you asking about the computational skills or
Yes.
stuff like that? Okay, yeah, it's really something. It's not a rocket science, Khalida. Is basically it's something you need to be trained or practice, or maybe someone has, because actually I spent like 5 years in Rothamsted research. So I was part of, I was member of a big molecular discovery, and my role was 80% actually looking at the data after it comes out from the instrument. Was a feature extractions and actually multivariate data analysis and nuclear networking and stuff like that. So I was basically focused on this. But of course, it's something you everybody can learn. Something you need some practice until you just get the wheel running. And then actually you'll be able, and there's guides everywhere. I mean, like some stuff you just, if you need, the problem is actually when you publish a metabolomic study in an international international journal, now actually reviews are much more harsh than before. So before, when the metabolomics just started, it was just basically we use as a natural product chemist, we use discovery, and we were not really interested or maybe paying attention to actually the rules that we have to use metabolomic study. I mean, like there's some guidelines. So I advise everybody wish to work on metabolomics to read these guidelines about, like, for example, the compounds and how you classify your compounds, level 1, 2, 3, and 4. There are 4 levels for the dereplication, and what kind of data would be accepted for this. And the thing is, the other thing actually, how you present your metabolomic data, and what actually the guidelines and lead you to make a successful metabolomic work, and that could pass the peer review process. So yeah, learn it, but should be learn the right way. Because one of the things actually I found that when we started metabolomic, because this was a field, we were not really aware with what actually the guidelines are. And then actually when I just spent is like few years now working in depth with the metabolomics. So I found like there are some guidelines we need to follow. It's not just old ways that we used to do, but there are some more other things we need to be careful for.
Okay. All right. Thank you, Ahmed. So the next question from Pranali Chowdhury. She asked, is there any bioinformatics role that involved in this process of drug discovery that you presented just now? Yes, could you please elaborate about this?
No, it's not. And there's it's me. I've done the work. I'm not bioinformatician. So I'm basically natural chemistry background, natural product chemistry background, but I've just learned more about the metabolomics. So it's nothing actually done relevant informatics or something like that. It's just just myself.
Yeah, because I think the one that you show is actually the molecular modeling, the feature extract from the mass, right? So you need to run MS to connect the molecular between that and see the similarities. It's just the software. Is it?
Yeah, it's a group of tools. As I said, there's a group of tools. Actually, you need to learn how to use them for your benefit. So of course, there are too many applications could involve these tools. But as a natural product chemist, you could use these tools in your benefit somehow. But you need to get trained and and it doesn't need really any bioinformatics skills at all.
Okay. All right, thank you. We move to the next question. Yeah. So this is from Muhammad Akil. Salam Dr. Ahmed, great presentation. I have some questions. The first one, can you enlighten what is the functional annotation in the data processing and you look into and the relationship with the data? And the second one, what is the tool you are using to do the correlation network with the MS data and how you implement Bioactivity into the network?
Yeah. So basically, the functional annotation process is you basically try to your samples. So, or you label them with different labels. So you can use these labels to correlate the spectroscopic data with their label you put on them. So for example, actually you could label the samples by like active and inactive. You could label them like liquid media, and you could label them actually this is so this soil is actually organic soil or inorganic soil, something like that. And actually, so these labels you put for actually would help you later on in the data analysis just to classify the samples and try just to find a relation between these two group samples. So for example, active and inactive, you have active and inactive, a group of active, group inactive, and you're using the chemometrics to find a correlation, or maybe not a correlation. You can find actually the molecules which is actually responsible for the discrimination of these two molecules from each other, or so discrimination of these two groups from each other. So this is actually the functional annotation step. So it's something you are doing in Excel. It's not something you actually need a specific software in it, but it's something you are doing in Excel. You have to functionally annotate your samples, give them labels, activity you are measuring. And for the second question, actually the correlation network, I use GNBS, which is some of you could be aware of it. It's actually a very open source platform. You can actually upload your sample there, and it will run a molecular network for you. And then you could actually download this molecular network file, and you open them in something called Cytoscape. So software for networks. You open this network in there. There's some parameter might be you need to change in the way how the network works. So you could actually look in depth network and see actually what molecules could be correlated together and see how you could use this information for your own case. How you implement bioactivity into the network. It's yeah, simply every network has like some data, and actually could be like MSMS or could be MS1. So basically you are adding another element to this table, which actually you, when you visualize the network, to take in consideration this extra elements that you put in, and you could see the correlation. Sometimes, I need just to be honest with you as well, sometimes some data, it didn't work with it. So it is not working with every data set, but we try our best. And when it works, it actually gave you a very, very good visualization for your data. If not, we could use some other tools. It's not only the same tool we're using in all research that we do, but sometimes actually the network could work, sometimes it doesn't. So we need just to use only analysis and multivariate data analysis. So yes, not every data set like the other one. So we use different tools.
So it depends actually with the data set itself, how it behaves, right?
Yeah.
All right. Okay, we move to the next question from Arif Zairi. Thank you, Dr. Ahmed, for the regarding the study on, I think, is it Aspergillus floccus? What are the bioactivity possessed by this Aspergillomarin B, fractionate or isolate compound from solid state culture?
Yeah, I didn't isolate or fractionate this compound. So I just said we didn't make further study and separate this compound because it's already identified from before. So what we found that these molecules is actually which is actually strongly correlated with the bioactivity, and we have seen this in the molecular network. And then searching this molecule we found a molecular formula or more than molecular formula, several hits. Some of them actually could be relevant to what we are doing. Them is actually not relevant. So this is what the chemotaxonomic filtration means. So you filter. So something, for example, you are working on Aspergillus culture. So it makes if this compound belonging to some fungi, for example. So this is how you filter your molecules, or you filter your hits according to actually to which class they are, or what's the source of them. So we found actually that these molecules, we found it in the database Aspergillomarin, and from the name is something coming from Aspergillus, and we are working on Aspergillus. So we said, okay, so let's see the NMR, the proton NMR, because isolate anything. Just let's see the proton NMR and see if we could find the peaks which could be relevant to this structure or not. And we were like lucky enough to see there are some peaks which are actually relevant to key peaks relevant to this molecule in the NMR spectra, and we to make sure from the identity of the molecule that way. So it's a known molecules. We didn't need to isolate it or anything by metabolomics, completely by metabolomics, that this compound could be involved or mediating for the biological activity of the Aspergillus flocculus. And this activity was anti-cancer activity.
All right, thank you. I think the example that you showed, type of compound, I mean, the workflow in metabolomics where you already know the compounds, and that is the point that you need to decide whether you want to isolate or you just, because for natural product chemists want to find the novel, maybe they just say, okay, stop, because it's already known. But maybe for pharmacologists for example, you want to find this, compound that, have this biological activity, then they maybe decide whether, they can buy the standards or they can first to get the compounds.
Yeah.
So it depends actually on, what is your purpose on your research project, objective.
Yeah,
So,
exactly.
yeah. So move on to the next question. This is the question actually from the Facebook from Dr. Go. Maybe you can repeat that. What actually the tools to construct the network with the pie chart? Because it's really interesting.
Yeah, this is I just answered that before. It's a INBIOSIS, an open source platform, so everybody can use, and you could go there. There is actually a documentation on every step. You can probably put you a link for this site actually here in the chat at the end of the question, and everybody can actually go there. You need to make an account just in case you need to use that one. And once you register, you would be able to find all the actually tools in there. Everything actually, there is a help. If you click the help, it will send you to the documentation. There is a step-by-step how to do what, how to do the process the network, load your data, how to convert them first, how you upload them, and when you collect your network, actually what you need to do with it, and all this kind of stuff. So everything is explained there. So I think in here in the chat, just in case anybody actually would be interested to use it.
Thank you very much, Dr. Ahmed, for that. If we move to the next question.
Yeah.
All right, okay. So we move to the next question from Dr. Ahmed Mediani. So thank you very much for the great presentation. It's very informative. For the molecular network, may I know which ion mode that you use? How valid is the data generated, and how actually you validate the data?
Yeah. So yeah, for the first part of the question, which ion mode? So you're talking about ionization. Yeah, I use positive and negative as a kind of a screening in the beginning, but actually when I and for the MS1, only the molecular weight. Sometimes actually I use only the molecular weight, not positive or negative. But in MS networks, I use the whether actually it's a negative or positive mode. So in the MSMS, I couldn't really rerun dual mode. So it's only one mode at a time. So it's probably most of the time I use negative ionization mode for our data. The validation of the data is basically, there are several methods of validation. So it started by actually manually looking and see actually how this actually looks like. And if you just stack several spectra together and you know the atom molecules you're already aware of from your work, probably on fungal culture, maybe if you are an expert in fungal culture, you would actually know that molecules could be actually coming, when you use the same method, it's just come at one retention time. So if you stack your samples together, fungal, see that these peaks is there in all of the samples. So you can see it by your eye. This is the first way of validation. But if we talk about further validator, there are several actually methods you can use. ROC, like this is one of the approaches actually we try to use to validate the data. And we could use also like some other things actually in multivariate data analysis, some statistical approaches actually to validate these data. And yeah, it's basically all about statistics. If you need to know if your statistic working well or not, so you use some statistical to just validate this data. If you need just yeah, is that Dr. Ahmed? Yeah, if you need yeah, if you need to speak about that later further, maybe, but it's more like about statistical kind of things. Yeah, but this is mainly the how do I validate the data.
Yeah, so basically the features in the multivariate data analysis, for example, maybe Simca, that
Yeah, there's
maybe
some, so yes,
can be used. Yeah, can be used.
yeah,
Sort
if
of
you're,
like to validate your.
yeah, if you are familiar with Simca, Dr. Halida, yeah, of course you are. But yeah, there are some like tools for the validation. So there's validation tools. One
This is
OC, VIB. You check the VIB for the
VIP.
features.
Yeah.
You actually check the if these features actually crossing the minimum limit or not. Yeah, I don't want just to go in deep with this one because actually maybe
Yeah.
some people will not be very interested actually in
That's
this one.
only
But actually, if
the.
someone is really interested in seeing what these tools could be helpful, I could actually, yeah, explain to that in details later on, specifically for Dr. Ahmed, because in a connection now, so we can have maybe further discussion about that if you want to.
Yes. Okay. Thank you, Dr. Ahmad. So we move now to the next from Dr. Sharul Nakasani. So the question is, what are the analysis tools that you use to correlate between the anti-cancer metabolite profile and the STIX activity in Euphorbia study?
Yeah, the same question. So the analytical, yeah, I use molecular networks to correlate chemical profile to the biological activity. So yeah, this is basically the tool I use.
So we move to the next question again from Dr. Ahmed Madiani. For unknown metabolites, what is your approach in identifying them? Any useful, easy way maybe?
Yeah, always identification is not the best part of the process and is always the most difficult,
Yeah.
the most difficult one.
Yes.
And easy way. It's more like there are steps you do, and if you are lucky enough, they will lead you to the structure. If you are not, you need to exert more until you find. Some people actually spend years to identify a compound, but it's yeah, it's basically if your compound is completely unknown and there is no hits in the database. One of the things could lead you to the structure is the molecular network. So for example, the molecular network is actually the MSMS is so the molecular networks connect the molecules according to the similarity in the fragmentation pattern. So if your unknown molecules connected to one known molecule, so you would know if they are structurally related. So you have an idea about the core structure of molecules from that way. So it's a very useful dereplication tool, the molecular networking. So it just leads you there. The second thing actually is, yeah, if it's NMR. So if you need to get 100% confirmation about the structure, your only way is NMR. I know sometimes it's difficult, sometimes the compound is not pure enough to run, or maybe the amount is not big enough to run NMR. NMR is not sensitive like MS, but yeah, if you need to have real confirmation about the structure, all you need to do is NMR. So when you do NMR, yeah, and with the other information you have got from before, from fragmentation, the molecular network, the database, that could give you an idea about the structure, how that could be, but the NMR would give you some confirmation about this structure. That simply it is.
Yep, I agree with that. Dr. Ahmad. But I think it's a very useful information that, you know, when we really get the unknown, and from the molecular networking, we really actually can see what's actually the containers, or the what are the similar compounds actually. So that from there, if let's say it's a steroids type of compound, how we would like to isolate them in comparison to glucose type of compound. Maybe we need to use polar column chromatography, sort of like that. So it will lead you to the lab, rather than, you know, last time in natural product we just use blind isolation without any guidance of it. But I think this is a good example, Metabolomics, showing you how to guide in formulation a new compound. So we move to the next question from Adi Sapura. So Salam Dr. Ahmad, how do you assign the left side in loading plot is active? I think it's in the multivariate analysis data, I think.
I'm just I'm not sure if I can get to this slide again.
Yeah.
Okay, here. Right. Okay. So you oh yeah, how could I assign? Yeah, this is actually the metadata I was talking about in the processing, the functional annotation. So you put labels for the samples. When you put labels for the samples and you run your BCA, these labels actually could lead you find, oh yeah, so this group here are active and these are inactive. So it makes sense. They are completely separated from each other. So yeah, this is this is what I was talking about, the functional annotation. So you put this data, you input the data, the active and inactive, inside your table, the one that you will send to the multivariate data analysis. Yeah. Mute yourself.
Sorry. So it's actually we adding some extra instruction to the software, right? So that they can, I mean, disable it and separate it based on what we're labeling.
Yeah,
right?
yeah.
All right. Okay. So the next one, I cannot see what's his name. Okay. Salam Dr. Ahmad. Kindly, I want to know you from your experience if we can make metabolic analysis for the data that collected from in silico without there any in vitro assay, or if there any correlation with the computer aided drug design or artificial intelligence in drug discovery.
I haven't really done that before. So the the correlation with the computer aided drug design, artificial intelligence, but there is something which I'm completely new to this part, but I'm trying to just some people to drag me to it. I mean, like I'm not expert, but I'm trying to learn it, which is actually because this is actually been very famous in recently with the COVID. So people actually they know they run this the in silico kind of things, actually just to give like scores for the compound if they are fit with the protein. I couldn't even remember the name of the approach. This slipped from my mind. But yeah, it's kind of like you are trying on a computer actually how this structure would be fit with your protein. All is a computer aided design, which some people trying to implement, but I'm not really into that yet. So I couldn't be your expert in here, but I'm really willing to more in this. If you have some experience in that and would like to apply in metabolomics, I'm really willing to collaborate.
Thank you, Dr. Ahmed. This is the next question from Nur Jannah. Doctor, I found only one database INPPAT, the Indian database to identify the phytochemical compounds for my plant. So is it okay for me to proceed using this database?
Yeah, any database. Okay, so some people actually, they built their own database. So it's like a built-in database, and you could use this database for the identification of your, there's, yeah, anything could be used.
Right, okay. I think we have the last question. This is a Facebook Live from Ronald Florel. Is GNPS actually a Windows-based database?
Yeah. It's, you can find it in on the internet. So it's, yeah, it's everything you do it online. So it's on the browser. On site, you upload the data, you run the network, and then you download your network, and then you work on this network offline. But actually, any processing happen on the GNBS is just happen online. So yeah, it's compatible with Windows, with anything. Yeah.
All right. Okay. Many questions asking, about the tools, how to do it. I think if in the future collaboration, I think what we can do is we invite you, Ahmed, as
Yeah.
possible. Then we will organize a workshop so everyone, can learn, because there are so many interests in this. So hopefully, just for the audience, stay tuned for the next project. I mean, the COVID is okay,
Yeah.
but if not, maybe we can plan on the online sort of the workshop, inshallah, because so many people asking how to, use the tool. I think doing workshop would be great. I mean, things to do so that they have hands-on doing this rather, telling, then they will remember.
Yeah.
So again, thank you, Dr. Ahmed, for your very interesting, informative, comprehensive presentation. You showed quite several type of examples that how metabolomics can be implemented in natural product discovery from different perspectives. What other you can incorporate in this metabolomics analysis. So just to update for today, actually we have about 106 audience participants that participate in this webinar, and also in Facebook about 30 participants. Very well done. Thank you, Dr. Ahmed, for your
Yeah. Thank you,
willingness
everybody, and I
for
will just copy and paste the GNBS now in a chat, so everybody can
yes.
just have an idea about that. Just give me a second. Yeah.
Okay. So while waiting, Dr. Ahmed, to type the link for the GNPS, so actually we have come to the end of the webinar. So again, I would like to thank to Dr. the very interesting talk, and I think it has opened up our view and give us an idea on how we want to bring our research output level.
Inshallah.
So before we say goodbye, inshallah, yeah. Before we say goodbye, I really hope that everyone can switch on our video so that we can have a photographic session together. And Ahmad, please do not leave after this, because we will be direct to the room so that I can have proper goodbye with you. Okay. Okay, I think, all right. So I think I just say 1, 2, 3, and then the technical will take our pictures. So everyone get 1, 2, 3. Smile. All right. All right. Intan?
Okay, done.
Okay. All right. Okay.
Thank
Done.
you.
Okay, thank you. Thank you very much, everyone. Please do not forget to the attendance form so that you can get the e-certificate and also the credit is as well for the UKM staff. And again, thank you very much, Dr. Ahmed, for your openness. I mean, to be one of our speakers.
Yes, thank you very much.
Thank you very much. Say, okay, bye-bye.
Alaikum
Assalamualaikum.
salam.